Can someone like pop in the chat and let me know that you can hear me okay? I'm using my office computer and I've not used it for Collaborate before.
I can hear you.
Great, thank you. Okay, so this morning we are going to be working through one of our recitation packets. But since this is something that we have absolutely not even touched on in class, and this is something we're going to do several times, what we are going to do is sort of work through it together. So I'm going to start by working through some new definitions with us, so we have something to base it on. Then I'm going to put us in breakout groups, which almost seems silly at this point since there is so few of us, but hopefully that will change. And you'll be able to work in groups, and you'll be each assigned a specific question from the packet. And so just so everyone's on the same page, we should all be starting on Lab 5 from our manual, which starts on page 31. And if it looks like I'm not looking at you guys, I'm just looking at my second screen, so I'm also following along on the same manual. So I'm specifically going to help everyone fill out page 32. And so there's a table for us to work on filling out. And so we can really have a good set of definitions for us to be working on to utilize. And so after that then is sort of, at this stage, kind of a semi-set of familiar questions. And so each breakout group, I guess, usually there's more of us. So at that point, each breakout group would be assigned a page. And so you'll have in your group some time to work on this and to think through, given the definitions we have, what answer makes the most sense. And then after about a half hour or 40 minutes or so, we'll come back. Your breakout group will be then responsible for presenting your answer to the whole group. I don't care if you're right or wrong. I just want you to try. And then as a class, we'll work through the question. Does that kind of make sense? Your silence, I'm going to go.
Yes.
Yeah. Okay.
Yeah.
So ultimately with these sorts of things, you've got 2 scores you're going to get. You're going to get a score for engaging and presenting. So everybody wants to do some kind of talking today. Again, it's not about being right or wrong. It's just doing a thing. And then your second score will be turning in all of the answers that you get today, right? So take notes when everyone else is talking. Submitting all of the answers that you get today into that same kind of quiz box that we're already kind of familiar with. Okay. Let's see. Be patient with me. All right, so I'm going to bring up a little whiteboard. Okay. So everybody can see a whiteboard now, I'm hoping. So as we look at the table, we find that ultimately, first and foremost, there are 2 groups of transport. When we're talking about transport, all right, transport in general, what we really mean is movement of materials across the plasma membrane. All right, and we've already teased about this in lecture, right? We talked about how ultimately you do need to get stuff in and out of the cell, right? And this was kind of this idea when we talked about that surface area to volume ratio. The cell needs to eat, right? Needs to get food in, the cell version of the cheeseburger. The cell's going to make cell poop. That cell poop needs to get out. So how is it exactly that we are going to be able to get those things in and out of the cell? Okay, so anything that we're talking about at this stage is sort of referring to all of that stuff. Okay, how do we get anything in and out of the cell? Okay, so transport in general comes into two groups. And that's going to be really the key at this stage. So passive transport is one group, and active transport is the other. And we can kind of see that in the table that we have. Passive transport has a lot of subgroups under it, but it's one big option. So we're going to start by focusing. Ooh, it doesn't let me edit that. All right, we're learning as we go. We're very excited. All right, so we're going to start by focusing on passive transport. Okay, so what are the things that make passive transport its own group? All right, so there's some key features with all of passive transport. Okay. The first and most important feature that sets passive transport aside from active transport, where's my text boxy thing? Let's get a different color here. Is we're going to do, ooh, that doesn't show up very well, does it? Let's not use green, I lied. Well, maybe? That goes okay, all right. Is anything that we're going to move using passive transport does what we call going down a concentration gradient. Okay, so what does that mean? Okay, I just want to click off. Oh, I clicked here. We'll get it, we'll get it. Oh no! I lost everything! Obviously, I'm very adept at this. Rats a frazzle. Put stuff back. Well, that didn't do what I thought it was going to do. Shapes. Rectangle. Okay, so here's my cell. If I'm going to do passive transport, okay, and I'm going to go down a concentration gradient, what that means is if I have a thing, okay, so say a sugar molecule, inside my cell, okay, I'm drawing, right, you're going to let me draw? No? Okay, apparently I have to make big swoops. Okay, I have very few of the item, and outside my cell, I have a lot of my item. Okay, so this is what I mean by a concentration gradient. I have a high concentration and a low concentration. Okay, everybody see that? So naturally, things want to move down their concentration gradient. Okay, and this intuitively makes sense, right? When you're in a really crowded space, especially now, okay, we want to move away from the crowds. Okay, when you walk into the DMV, you don't want to sit where all the people are sitting. You want to go find an empty seat that has like 3 other empty seats around it, right? Okay, and that's all we're seeing here is we want to move away from the crowded space into the more empty space. Okay, so this is what passive transport means, that we're just moving naturally away from the really crowded space into a space that has more elbow room. Okay, and this is passive because it's something that we all want to do, right? It's a natural mode to do. With me so far, this feels okay? All right, I'm going to assume if everyone's going to be quiet that it feels okay. So if you have questions, you either need to turn your mic on because I can't actually see you. You can raise your hand, I think, or you can pop into the chat, I think. Okay. All right, so the other key feature then, because this is something that is a natural prospect to do, right? This is something particles want to do. Am I texting again? All right. Is that this does not cost ATP. Okay, so remember we learned yesterday that ATP is the currency of the cell. This is a free, so to speak, process. Okay, feel good so far? So you're not gonna give, I don't have an actual eraser. So every time I want to do something new, you're gonna make me erase the whole thing. Fine, I'm not gonna complain. Okay, so under passive transport, we see on our table that there are 3 subtypes. Okay, diffusion, facilitated diffusion, and osmosis. So this is all going to be variants on a theme of these molecules wanting to trend away from these really high concentrations. Okay. Now the other thing I want to point out is you see on here this box that's in orange on the page that's talking about equilibrium. Okay. So that's asking the question, as we're trending away from high concentration to low concentration, how many of my green particles are actually going to move over? Okay, apparently my cell is black now. That's fine. Psst. There we go. Okay, so equilibrium is the state, as the name implies, where you have equal amounts of your particles on both sides. So enough of these green guys has moved over so you don't have a high and a low anymore. Okay, now as you'll find, however, and as is the state with people and with everything else, okay, that doesn't mean that everything stops. Okay. Oops, that's not how you spell equal, is it? Okay, so all this means is we have equal amounts on both sides. Now our particles are still moving. Okay, one of the rules you want to keep in mind is a stopped cell is a dead cell. Okay, so our particles are still, that's very annoying. Our particles are still exchanging from one side to the other, but it's equal and random, right? This guy is going to move to this side, this guy will move to this side, but there's no concentration gradient anymore, right? There's no side that has a really crunched up, smushed amount. So there's no net, what you'll see your book called net movement. There's no movement where everybody's going in one direction anymore. So this is equilibrium. Okay, so this is the goal, right, is to get the same amount of individuals on both sides of our plasma membrane, which is what the lines in my box are representing. Okay, this still feel okay? Silence says yes?
Yeah.
Good. All right, I'm going to clear this off then. I am recording this, so this will be available on our lab page for later. Okay, so let's start looking at the 3 different types of passive transport. First up on the docket, the smallest and most simplest, diffusion. All right, so what sets diffusion apart from the other 3 types of passive transport? If it's still passive transport, we still have to go down, whoops, a concentration gradient. Okay, it still has to be free, so cost no ATP. Okay, but that doesn't make it special. That just makes it passive transport, right? So what makes it different from the rest? Okay, so diffusion, here's our cell. Here are our green dots. Well, I guess they're circles now. These are squares. We're doing good today, right? Okay, so there's our concentration gradient. Remember, we're going from high Hello. Okay, so all of this is still the same. So what's different? The key feature that makes diffusion different is the type of molecule or solute that's actually going to pass over. So diffusion is the most simple version of our three. Okay, simple in that it is only going to use a specific subset of molecules. Go ahead and leave that in red. So diffusion is only going to use small non-polar / charged molecules. So it's picky. Okay. So examples of this would be things like oxygen, right? O2. Carbon dioxide, CO2. Okay, so for the most part, diffusion, oh my gracious, please stop. Oh, look, I have a pointer. Okay. Diffusion is the process by which cells breathe. Okay? Cells need to breathe as much as they need to eat or anything else. So these very teeny molecules can get through our plasma membrane. And this kind of makes sense. If we remember that our, wait, I hit my lips. Our plasma membrane is made up, nope. Oh dear. Can I move this? I can move things. Look at all the things I'm learning. Okay, our plasma membrane is made up of these phospholipid bilayers. We're going to pretend these are lined up a little better than they are because this is extraordinarily difficult. Okay, so these are very close together. Okay, and the tails, being nonpolar, do not want to let things pass through. So if we are going to squeeze through, okay, as a teeny oxygen model, okay, I'm very small. I'm nonpolar for the most part. I'm not charged, so I can squeeze and get into the cell from out of the cell. That works because I'm pretty small. All right, and so no one's going to notice me enter. Okay, but okay, if we remember our big glucose rings, this actually should be an O. Okay, this thing is a chonquina, right? So if this thing wanted to slip through, there is no way that thing is going to fit, right? It's too big, it's too polar. Hey, there's no way this thing is going to be able to fit through these tiny holes. So it's going to need a different process. So diffusion is extraordinarily picky in that it's really going to only fit these small, oops, this should be non-charged. My bad, guys. Not going to let me edit that right. Non-charged, non-polar, non-charged molecules. Okay, so very discrete, non-exciting things that are going to squeegee or shimmy. Okay, we're going to imagine things shimmying their way through these teeny weeny spaces. I want this to still go down that concentration gradient. Okay, so in this way, we have filled out the first column of our box on page 32. Any questions about diffusion or passive transport as we've identified it so far? Okay. I don't think that's still spelled for facilitate. Yeah, I got real excited there, didn't I? There we go. Diffusion. Okay, so let's look at the second box then, facilitated diffusion. So we still have a bunch of the rules. We're still under passive transport, right? So we have to go down the concentration. All right, great question. So in, more than one person. Nope, just, okay. Abigail asked, does Equilibrium use ATP? So it does not, right? Or it shouldn't, as long as we're under passive transport, right? So nothing that's happening under passive transport uses ATP. Okay, so as long as we're reaching equilibrium under passive transport, reaching and maintaining equilibrium will be free. Great question.
Is facilitated diffusion and diffusion the same thing?
So they are not, and that's what we're about to tackle. So they're both passive transport, but there's small differences in what the types of molecules they're going to allow. Okay, so diffusion is a type of passive transport, but it's only going to allow little molecules across. We're still going down concentration gradient. It's still going to be free with the goal of reaching equilibrium, but it's only going to let those baby molecules across. Facilitated diffusion is going to allow us to tackle those fat molecules, but in order to do so, we can't just use this teeny space, right? We have to have something else. And that is not the word concentration. Oh. Any other questions about regular? I can't consent. You know what? You guys get it. It's fine. Okay. Any other questions about regular diffusion, equilibrium, or passive transport? And we're going to focus on facilitated diffusion in a second. So hopefully the comparison of the two will be very clear in a hot second. Okay, great question. Okay. I clicked the little rectangle button. Don't be rude. I'm going to make this one a little bit bigger. Yeah, that's fine. Okay, so here's our concentration gradient. Okay, so all the other, oops, text. All the other rules are still in place. Okay, but now, okay, before we were actively excluding very large molecules, or perhaps molecules were charged, right? So for example, our glucose molecule was unable to pass through, right? We only let in things like oxygen and carbon dioxide. So this begs the question, we know that things like glucose or even bigger polysaccharides need to be able to get into the cell, right? Our cell needs to eat. So how does that happen? Okay, so this is where facilitated diffusion comes in. Okay. Draw me up a little. Yeah. Oh, maybe, well, it's fine. It's just a very, that one's very big. I won't waste too much time on this though. Did not leave myself a lot of space for my phospholipid tails, but you guys know what I'm drawing. Okay, so if and I'm not going to redraw this, it was just going to cross my line here. If I want to bring a glucose over, right, or very large particles, I still can't squeeze through the holes, right, or these tiny, tiny spaces between my phospholipids that make up my plasma membrane. Okay, that's still a problem. But I do need to get the stuff across, right? A cell gotta eat. So how do I do that? Okay, it's time to bring in the big guns, aka our favorite biomolecules. All right, so what facilitated diffusion does is use, very poorly drawn, protein channels. Yep, that's almost on there. So protein channels, stop it, work a lot like tunnels or bridges or gateways. And these tunnels and gates, text, allow larger molecules to pass across, okay, using secure checkpoints, but that are also big enough to get across. Okay. It also allows some smaller things to get across much faster. Okay, so now things like glucose, okay, and other sugars or food can cross. We can move that so it's not sitting. No, not you. You. There we go. Okay, so this is the big difference between these two, right? When we're looking at diffusion, we're only moving small things. We don't need to modify it. We don't need to use a security gate. When we're using facilitated diffusion, the facilitation, right, is really talking about a modification. In this case, our protein channel. And this protein channel then acts like a security gate or a security tunnel that allows us then to move much larger items across our tiny, tiny spaces much easier and much safer. Okay, all the other rules still apply, right? We're still moving down a concentration gradient, okay, but we're doing it in a way that's safe and easy. Okay, the goal of this and the difference between these two, very clear?
Yeah.
Okay. Obviously, I need to erase this to talk about my third one. So, are you guys okay with that? Like I said, this will be erased, but do you have everything you want, or this will be recorded, but do you have everything you want for the time being? Okay. Okay, so let's talk about osmosis. Osmosis is probably of all four of these the most complicated and the most confusing often for students. So all of our other rules still apply, right? We're still ultimately working down a concentration gradient. And this has no ATP. Okay, but osmosis is particularly unusual in the way it works. Okay. So osmosis is sort of like the last option when you're really trying to get to equilibrium. Where am I? Okay, here's my setup, same setup we've been using. High concentration. Here's my low concentration. I'm starting to get this drawing thing down. Okay, so we only tend to utilize osmosis, or cells only utilize osmosis when we have particles that are so big, so charged, or so polar that they really can't move across our plasma membrane at all. Okay, so we can't use regular diffusion and we can't use facilitated diffusion to reach equilibrium. Okay, and that's because osmosis in this case isn't the movement of particles like the other two have, right? It was always the movement of glucose. It was the movement of oxygen. So it was always these boxes of things that were moving around. In this case, it's the movement of water. Okay, so it's actually going to be moving the fluid around. And this is a lot for us to kind of wrap our head around. So if we imagine back, oh my gosh, that's so annoying. If we imagine back to that DMV, okay, or that classroom example we had, but how you never want to go and sit in the space where everyone's already sitting because it's really cramped. Okay, that's still the issue, but at this situation, right, it's like being stuck in a space, right, where the DMV is already super full and there isn't any way for you to get up and move. Okay, so that's the situation we're in. So instead of you getting up and moving, right, so we're not moving. Instead, we're going to bring in more chairs. Okay, so ultimately, the goal is the same, right? And the result is the same, because if we bring in more chairs, we're still going to be able to spread out everybody. Okay, so the concentration of individuals is going to be the same. We're still social distancing, right? So everybody's still going to have the same amount of space that they had before. All right, let's go ahead and draw that. Nope. All right, delete you. Oh, there we go. Now I figured it out. It's the greatest thing since sliced bread. I have lost track of how many of those things I have, so let's say it's the same. Okay, so reaching equilibrium here and getting to equilibrium here looks and feels a lot different. Okay, the goal is still, gracious, okay, that everybody has an equal amount of space. Okay, but we didn't do it by picking up and moving these guys so there's an equal number on each side of the barrier. We just want everyone to have an equal amount of space or an equal amount of elbow room. Okay, so what we've done is move chairs, okay, in this case fluid or water, to the more concentrated, right, side of our plasma membrane, whichever side that might be. Okay, so if we got this, for example, on a test question, you would see it specified that sucrose, right, or this positively charged ion cannot pass the plasma membrane, right? How would you fix that, or what would happen? Okay, and this is one of the few ways in which you can fix that is by taking water and moving it over. Does everybody see what we did here? This is, in my opinion, one of the harder ones.
So when it's in the cell and outside of the cell, can they still move across both sides?
Can what still move across?
Like the water molecules, like how there's an arrow pointing out on the diagram on the far left. There's an arrow pointing out but not in. So can they move out but not back in?
You're talking about the water?
Yeah.
So in this case, the water is moving out because its goal is to reduce how clustered or over smushed everybody here is. So that's the only reason the water's moving out, right? Is because all these guys are way too cramped. And so that's just the goal of osmosis, is we want to decrampify how smooshed everybody is here.
When you're doing that, can things go back into the cell?
So, and that's what we see over here, right, is once everybody is decrampified, then we have water moving in both directions.
Okay. So
absolutely. So it's just while we're decrampifying, we're working on fixing that concentration gradient. We want to de-smoosh or decrampify.
Okay.
And if this was the opposite way, if we had our high concentration inside the cell, then you would see that arrow facing the other direction. This just happens to be the way we were drawing it, so I kept the direction the same. That answer your question clearly?
Yes.
Okay. Good question. Any others about osmosis or any of the other, because this is all of passive transport now. Okay. All right. For the very last one, active transport, I'm going to have to erase the board one final time. Does everybody have what they want off of my drawing? All right, I don't know why I'm typing back. Too much from lecture. So Karina's question is, she's asking or assuming that these types of transport can happen simultaneously, and she's absolutely correct. All right, so depending on what types of solutes or molecules are being moved across the cell, so we think about diffusion is moving across oxygen, facilitated transport is moving across food. So certainly at any given time, right, a cell is going to need all of those things potentially simultaneously. So what is occurring is just certainly dependent on the needs of the cell. Now, I'm never going to ask you a question that's probably going to ask that you do all 5 at once or all 4 at once, but you may need to do maybe more than one at a time, but after you've done some practice. None of your questions today are going to probably make you do more than 1 or 2 at a time. Okay. Good to clear? No cries of injustice? Okay. So let's take a look at our very last. Okay, and so now we're moving over into an entirely different category, active transport. So now, up to this stage, we've had passive transport, which has all had a very specific subset of rules that make that its own category. So now that we're moving into active transport, pretty much all we need to do is flip the script. So passive transport always move down concentration gradient. Active transport, we are moving up a concentration gradient. And I really can't spell that word, can I? Constant. Oh, I'm missing the other N. Good thing I'm recording this. That won't be embarrassing later. Right? If we're going to move up a concentration gradient, right? So here's our cell. Right, so I'm actually going to flip the way I'm drawing this to make it more interesting. Okay, so now you notice that I have a low concentration outside of the cell, but a high concentration inside of the cell. Okay, so I would be moving up concentration gradient if I wanted to move something inside there. Right, if I'm going to do that, this is going to cost ATP. Okay, dollar bills, yo. Okay, so those are the big keys. Now, it doesn't matter what molecule at this stage I'm moving across. If it's oxygen, if it's sugar, if it's charged ion. So most commonly, a good example of this, so sugars do this. We like to bring food in. And we want as much food as possible. Commonly, we might see charged ions like calcium. Calcium is one of the ions we use to make our heart pump. Okay, so certainly we need a lot of those. We need to keep our heart going at all times. Okay, other charged ions like sodium. Okay, we use those to make our muscles contract. Okay, so we usually have a plethora of those types of ions. Okay, so we always have really high concentrations of those in our cells. So anytime we need to bring these in higher concentrations in our cells, we need to use active transport. Okay, but it doesn't matter what we're bringing in. Absolutely, equilibrium or its concentration gradient. Because it may not even be that these are equal, but it is even higher, right? So it costs ATP to go above and beyond, and you're exactly correct, right? That's what's making it charge money. Okay. So if we're going to push higher than where it is on the side we're coming from, that costs money. Even if it's a few, right? If we're going to push past equilibrium, that costs money, or ATP. Okay, and on the plus side, active transport is that simple. That's all it is. Okay, there's no fancy business, there's no nothing. That's all it is. Okay, the key feature is we're going to have to pay a toll. Okay, now oftentimes with active transport, if I remember using sugars or these charged ions, right, I am probably also using protein channels, etc., to get them across. Okay, but the key feature is I'm paying to do it. Any questions about any of that?
Can oxygen or any of the smaller non-charged molecules, can they be a part of active transport?
They can. It's less likely because things like oxygen and carbon dioxide just tend to be used up much quicker, but they certainly can be.
Okay.
Good question. Anything else? Okay, how many? Let's see, there's 16 of you. And we have 1. Okay, let me do some hot math really quick and figure out how many groups I need. Okay. So if I did 4, that would put 4 people per group. So let's do that. And then, okay, so here's what's going to happen. I'm going to put you guys in breakout groups, which just means that as soon as I hit go, you guys are going to be put in small groups. The group number that you have is the question that you have, right? So group 1 is going to do the first question, or page 33. Group 2 will do the second question, or page 34. I'm going to check in on you and make sure you know what question you're doing, so don't worry. And so basically, once you get in your groups, you should be in groups of 3. I don't think I have it set so nobody's doing the last question because it's the hardest. So we're all going to kind of work on that one together. It's not the hardest. We haven't touched any of the information per se. So you're the least prepared for it. Let's put it that way. What I want you to do is look at the question you have in your groups, read the scenario quite clearly, because it's going to set up which molecules can cross the barrier for you. So it gives you sets up the scenario quite clearly for you. And then read the option answers. And so what you'll need to do is tell me which answer is the correct answer, right? And so that one's fairly straightforward, so tell me which answer is correct. But then for each of the wrong ones, tell me what's wrong with it. And it might be as simple as, well, you would have had to have paid ATP for this, or this isn't moving down its concentration gradient. So think about what's wrong with the wrong ones. You can use every note we've taken as your information source. Don't forget that we have our textbook, which is online and easy access. You should be in chapters 5.2 and 5.3, I think, which are just simply passive transport and active transports. They're very clickable. They're very searchable. You can just go into the search bar and say diffusion, and it'll pop up for you pretty quick. So don't hesitate to use that either. I'm going to give you about 20 minutes to a half hour, and I'm going to kind of poke through, drop myself into your groups and check on you to see how you're doing. And then after that time, I'll pull you back out and you can present to the group what your answers were. Okay, any questions about what you should be doing at this point? If you have questions, you just need to have one person in your group do the raise hand thing so I know to pop into your group. Or I think the chat feature still works too. Okay, and you'll obviously need to use your mics in your group. Okay. Make sure everybody got a group here. Sounds good. Here we go.